is what we call sequencing. What is a genome? Genome basically represents one full complement of genetic material in an organism or a cell. And sequencing the genome has been challenging. Why is it challenging? We'll come to that in a moment, and we'll get into more details as we move into the talk. But the question that is very basic is, why do you want to sequence the genome in the first place? Why should we basically bother to sequence the genome? One is that, as you know, DNA is the blueprint of our existence. So we must know what is there in the code. So we were interested in knowing what makes us human. How do we compare with other organisms? What is the difference that makes us human as compared to chimpanzee or a bird or a frog and so on and so forth? Then we wish to identify variations, which are both intraspecies and interspecies. And particularly for biomedical applications, we are interested in intraspecies variations. And more importantly, we are interested with intraspecies variations that associate with disease. Because once you know which mutation is causative of the disease, only then can you go into the further biomedical interventions to address the disease. And if you are reading currently, you would be knowing the revolutionary technologies that CRISPR-Cas have come in that can edit the genome very specifically at a specific position, allowing you to remove the mutation that causes an inborn genetic disease. So what are the steps in genome sequencing? So first thing is that while we would want to sequence a genome starting from the 5' end until the end of the chromosome, it is not possible because sequencing technologies have a limitation. There is only to a certain length that you can read before you cannot read beyond a certain length. So therefore, when you're sequencing a genome, first thing that you need to do is to fragment the genome into smaller pieces. You fragment your genome, and then depending on the technology that you're using, you will have specific size fragments, at least not bigger than the size specified. Sequence these individual fragments together, and then once you have these fragment sequences, you need to assemble them together back into the genome in the order they were in the genome. So you assemble it together back here and get your full genome sequence. Right? So if you look at these steps, you have to create a genomic library, basically split the genomic DNA into smaller pieces. Then fragments of genomic library have to be sequenced, and then you put them together in the order they appear in the genome. So these are basic steps in genome sequencing paradigm. Now, another very important concept in sequencing is read length. So for example, here, if I give you a puzzle here, this is an original image that I've clicked while I was in Paris. And then if I tell you that you can fragment it into 4 parts, 9 parts, and 56 parts, and then it has to be reassembled, then it is very easy to see that the one with 56 parts is most likely and most difficult to be assembled. And also it is error prone. For example, this portion can very easily be placed here, and this portion can very easily be placed here. So you can have errors in assembly here. The smaller the number of parts, the easier it is to assemble. For example, here only 4 parts. So this is very easy. You cannot go wrong here unless you have absolutely, you have gone crazy. So if you see, this is your original genome. This represents your genomic library. These are your long reads. The longer the reads, the lesser number of reads you need to assemble to get the original genome. The shorter the reads, the more you have to assemble, and the more you'll have problems in assembly, and you can have errors. And more so with respect to human genome, because in human genome, you have 50% repeat content, which means the reads are, these reads will be present in the genome at multiple locations, and you'll not be able to identify correctly where this particular sequence that you've got will assemble. So basically, read length is important. Read length, how do you define the read length? Read length is defined as the maximum sequence length that can be obtained in one sequencing reaction. And this is specific to each of the sequencing techniques that you use, and we'll go through how this varies between the sequencing techniques. And as I told you, it is a critical parameter for the completeness and accuracy of the genome sequence. And more importantly, with respect to genomes that are with high repeat content, and sequence the human genome in 2001, it is still not absolutely complete. It is still a working draft. There are regions which we do not know, and there are regions which have misplaced, and we are still working on it. And that is why if you go to the UCSC genome browser, you would see the assemblies have been revised over time. Then another important concept is depth or coverage or X value of your genome sequence. So for example, here, once you get your reads, you'll align it back to the reference genome. For example, at this position, chromosome 5, position 500, you have 8 reads aligning onto this position, which means the coverage for this position is 8x. At another position, let's say chromosome 500, only 2 reads have got aligned, which means the coverage at this is 2x. Then you have gaps here in between where there are no reads, and then you can have other positions where you can have more or less coverage. But on an average, you can also cover the, you can also say what is the average coverage of the genome by using the formula here, which is average coverage equals to total number of reads into the length of smaller reads divided by the total genome length. So if you see here, total genome length is fixed. The length of the individual reads is also fixed more or less depending on the technology that you use. So all that you can do is increase this N. You can sequence a single position multiple times, so has to have a higher coverage. And this higher coverage is important because it gives you a higher statistical power and confidence in the analysis. This is a very important concept, and why next generation sequencing techniques are scoring much better than previous techniques is because here you can have very high coverage, as much as 50x, 100x, 200x, depending on how much you want to invest in your sequencing expenditure. You can have as much coverage as you want. Then coming to sequencing techniques, most sequencing techniques are polymerase based. So you have DNA polymerase, it will synthesize a complementary strand. And this complementary strand, while it is adding nucleotides, will record which nucleotide gets added. Then there is also one technique that uses ligase-based method. This is called solid. It is now out of use, and we'll not discuss this for today. But then just to remember that ligase has also been used for sequencing. And then now we have a sequencing technology called Nanopore, which uses helicase-based method. So helicase, if you remember the DNA modifying enzymes, helicase is basically to unwind the DNA. So you unwind your DNA, and while it is unwinding, you read and you get the sequence. So here is the basic polymerase-based sequencing reaction. So you have a template here with a single stranded. You have added an adapter at one end so that you know the sequence here, and to that you add a complementary primer, and then you give your DNA polymerase here, which will extend this primer and add complementary nucleotides to this reaction. So basic reaction that you do here is this primer is represented as DNA of length N. You add your DNTPs, you have your DNA polymerase. Now, when you give your DNTPs, that your DNA will get extended by 1 nucleotide because they're giving all 4. And alongside it, it will release a pyrophosphate, and it will also release a hydrogen ion. Right? This is important that what is released, because each of these is used as a reporting molecule for the sequencing reaction, and we'll see how it is used as we move into the technologies. So here is your reaction. So you get your T, A, C, G, T, T, and based on what is there on the template, you have your complementary nucleotides added. Now for the sequencing reactions that use polymerase, most commonly what you use is a dideoxynucleotide triphosphate. Now dideoxynucleotide triphosphate is basically a DNTP whose 3' OH has also been removed. So this is, there was no OH here, there's also a 3' OH removed. So now this cannot be extended any further by polymerase. So once a DDNTP comes and joins, now this fragment cannot be extended any further. So therefore, if you remember your Sanger sequencing and the dideoxynucleotide, how they're used, and each of these dideoxynucleotide is differentially colored. So A will have a different color, T will have a different color, C and G, and so on and so forth. So if this is your reaction here, and you give your dideoxynucleotides to the normal nucleotides, what you'll have over the course of reaction is a series of fragments that differ from each other by 1 nucleotide, and the last nucleotide will be your dideoxynucleotide in your mixture. And because each of the dideoxynucleotide has a different color, you can now do a size separation. And once you do a size separation, you can record the color that is emitting from your particular nucleotide, and that will give you the sequence of the nucleotide. Am I audible and clear? Anyone?
Yes, sir. Yes, sir. You are clear.
Great. Okay. So in automated Sanger sequencing, we use differentially labeled fluorescent DDNTPs. This facilitates resolving of fault fragments. Then you would use differential labeling, as I told you, so you know which nucleotide is coming based on the color. Then you use capillary electrophoresis, which gives you higher resolution and faster run. And then you can do 96 samples in one go of the run, and you can place 4 such runs together. And then the sequence output that you get here is in the form of a chromatogram. So you get these colored peaks, and based on the colored peaks, you can decide which is the nucleotide at that position. Here, initially at the start of the reaction, you'll have some confused peaks, which is a property of the technique. But as we move on into the 30 odd nucleotides, it starts getting clearer. So automated Sanger sequencer was one of the most common used sequencers until 2001. And major achievements for this sequencer include the sequencing of the Haemophilus influenzae genome, the first free living organism to be sequenced. In 1996, we sequenced yeast, 2000 Drosophila and Arabidopsis. And then the crowning glory of Sanger sequencing was that with this sequencer, we were able to sequence the human genome in its completeness, almost completeness, so as to say. However, the sequencing of the human genome also brought out the fact that if this is the technology that is to be used, it may not be possible to sequence all individuals over the time. Because if you see, sequencing the human genome, it took around 10 years and $3.2 billion. So the cost was prohibitive and the time was equally prohibitive. We want to move into personalized medicine where we want everybody to be sequenced before a medicine is administered to him. And I'll give an example later in the talk why it is important to know what variations you carry when you go to the doctor. And so what was required was a faster and a cheaper sequencing technology. And that is when people started looking for the next generation sequencing technologies, or the second generation sequencing technologies, as we call it today. So we are looking for speed and cost effectiveness. The challenge was to produce a technology that can sequence a human genome in around $1,000. Right. So based on the timelines, you have currently 3 generations of sequencing technologies. Some people call it 4 generations as well. So from 1976 to 2002, you had Maxim Gilbert and Sanger Coulson. Sanger was the one which was most common. Maxim Gilbert had its limitations, so was not so much used. Then second generation started around 2002, and some of the technologies are continuing in late, and some of them have become now redundant. For example, PARO-sequencing, as you see here, is out of phase now and is not being used. Virtual terminator sequencing is currently the most common and commercially most viable sequencing technique that we have as of today. Solid also is now out of use. And then over the period of time, we have moved into the 3rd generation sequencing techniques. And now we are finally into Nanopore, Ion Torrent, and SMRT, or single molecule real-time sequencing. All right. So now I'll take each of these techniques one by one. I will not take solid and pyro sequencing because these are already out of use, but in case somebody wants, you can write to me and we can discuss later. But we'll discuss virtual terminator sequencing, Nanopore, Ion Torrent, and SMRT for the purpose of the talk today. So in 2009, if you see, this is a review from around 2009, and you can see the read length, and at that point of time was 250 base pairs for pyro sequencing, 36 base pairs for Illumina, and 35 base pairs for solid. And over time, there have been increase in the read lengths, but only to a certain extent. So you now have around, for pyro sequencing, 400 base pairs. You can have 100 base pairs for Illumina and around 60 base pairs for solid. But of course, these are now redundant. And now you have a new class of sequencers, which we call long read sequencers. Now, as compared to short read sequencers, virtual terminator sequencing, Ion Torrent, which give you around 200 to 100 base pair of sequence for one sequencing reaction, Nanopore and SMRT can give you much longer sequences to the tune of, say, kilobases, 20 kilobases, 30 kilobases, or even more. But on an average, around 20 kilobases, 30 kilobases is what you get from here. So we'll go into these technologies, how they enable long sequence reads possible, is what we're going to discuss later during the talk. So some of the common features of second generation sequencing techniques. Number one, you have to prepare a library, as I said. So you have to do a random fragmentation of the starting DNA. You have to add your custom linkers so that you can add primers that can amplify these regions. You do a library amplification, and library amplification can be done depending on what is the platform that you're using or the sequencing technology you're using. You can either do a bridge PCR or an emulsion PCR. Then there is direct detection. As soon as the nucleotide is incorporated into the sequencing reaction, you get to know which nucleotide got incorporated, as compared to Sanger sequencing where you had to run a gel before. So that was not real time. This is real time detection. And more importantly and significantly, this is massively parallel sequencing. You can do 1 million to 1 billion samples in one sequencing reaction of the machine or one instrument run, which is the major strength of the second generation sequencing technologies as compared to what you got in Sanger, where you could do at max of 96 samples and four plates, so which means around 3 to 6 samples in one run of the machine. But here you can do millions of samples. So while the read length here is around 100 to 200 bases, because it is massively parallel, the sequence output is high throughput. In Sanger, by the way, the maximum read sequence was around 850 to 900 bases. And then, of course, these are short read lengths, shorter than Sanger sequencer. Sanger sequencer gave you around 900 to 850 bases for one sequencing reaction. Here you get at max around 100 to 200 bases, as I told you earlier. So this is a virtual Terminator sequencing machine. This is sold by the company called Illumina, and the trade name is called Solexa. So all three are the same. This is basically a polymerase-based sequencing reaction. So it is very similar to Sanger sequencing, except for the fact that here, instead of DD and KPs, what we use is a virtual or reversible terminator. So what is a virtual or reversible terminator? This is a nucleotide that has a fluorochrome attached, and it also has a virtual terminator moiety attached. As long as the virtual terminator moiety is attached to the nucleotide, no addition of new nucleotide can happen. But then you can chemically remove the virtual terminator, along with the fluorescence will also go. And now for the same reaction, you can extend the same molecule further down. So in contrast to Sanger sequencing, where once a dinucleotide got added, it cannot be extended any further. Here, you can stall the reaction, note down which nucleotide got added because you have a differentially colored fluorescence here for each nucleotide, and then remove the termination and allow for the extension of the same molecule and add the next nucleotide. Right? And the termination, the reversible terminator, you can basically remove by using tris carboxyethylphosphine and other chemical substances, depending on what technology you're using. These are some of the reversible terminators that are commonly used in Solexa. And there is another company called Helicase, which uses slightly different terminators. But then the most, as I told you, the most commercially viable is Illumina or Solexa. So here is what is happening here. This is what we call a flow cell. A flow cell will have millions such templates, and each template will be in multiple copies around a local area. Here I'm showing you 2 different templates. Here is your sequencing primer. You give all 4 nucleotides together. Who is drawing here? All right. Let me see how I can remove that. Did I draw that? No, somebody else did. Does it come to the next nucleotide? Okay. Please don't do such silly things here, please. This is the class, maintain the dignity of the class, right? All right, so if you look here, this is your template here. You gave your first round of reaction. Since all 4 nucleotides are colored, you can give all 4 together. Depending on their base complementarity, the nucleotides will get added here. And this represents your virtual terminator knob. So as long as this virtual terminator is there, no further addition of nucleotides will happen. So you can now note down what is the fluorescence at this position here in the flow cell, what is the fluorescence at this position here, and that gives you the nucleotide that got incorporated. Next, you remove the virtual terminator moiety. Now you're ready for the next round of reaction. Again, your next nucleotide, and so on and so forth, right? Can you remove this red line, whosoever has drawn this? Please don't do such silly things here. There has been some effort. Some effort has gone into making these slides. If you are not good enough, please leave the talk and move on. But don't disturb others. As students, I do not tolerate such indiscipline. Please don't do this. Thank you. All right, so let's say here we are looking at the flow cell and we're looking at two positions here. This is position 1, this is position 2. In the first round, you have green fluorescence, which means the cytosine has got added here, right? On both the positions, which means on the original template, there was a guanine. Next cycle, you have a blue here, which means an adenine got added here and still a guanine got added here, so on and so forth. Now you have a thymine getting added at one position, and this is remaining green, which means there's still a continuation of guanines in this sequence, so on and so forth. You can keep doing. And as I said, the length that you get here is around 150 nucleotides. So for 150 reactions, you keep extending the nucleotides, and then finally you get your sequence here. So if you see position 1, this is the sequence based on the color that you observed in these positions. And for the second position here, the bottom, you have a continuous stretch of cytosines for 6 nucleotides, and so on and so forth. So this is basically Solexa or Illumina, massively parallel, a read length around 150 bases now, and the most commercially viable and successful. And the idea is that Illumina is also customized. If you want high throughput, you can order a different set of machine. If you want slightly lower throughput, you can order a different set of machine for your reaction, for your projects, right? Now we come to the next sequencing technology, and this is Ion Torrent. Now, Ion Torrent, again, is polymerase-based sequencing technique. So you have your ddNTPs, and here you don't add any color. You know beforehand which nucleotide you have added. You give only one species of nucleotide, and then you see whether the reaction is happening or not. How do you record whether the nucleotide got incorporated or not is via checking the acidity of the reaction. So whenever a DNTP gets, whenever a piece of DNA or DNTP gets incorporated and DNA gets extended, you'll have a release of hydrogen ion. The release of hydrogen ion means you'll have an increase in the hydrogen ion acidity, in the acidity of the reaction, or a decrease in pH. So instead of checking for the fluorescence, here you read the reaction or incorporation by checking whether the reaction acidity has increased or not, or the pH of the reaction has gone down or not, right? So this is not imaging based as compared to Illumina, which was imaging based purely. So upon incorporation of complementary nucleotide by polymerase, a pyrophosphate and an H+ ions are released. The release of H+ ions serves as signal in Ion Torrent. So what you have at the base of each reaction is a pH meter, a digitalized pH meter that can record changes in pH, and that allows you to know whether a nucleotide got incorporated or not. Difference from virtual Terminator sequencing is that here in one reaction, only one type of DNTP is given. So you know that you're giving a guanine, and then you see in the flow cell which spots showed a decrease in pH, which means there in those spots at that position was a cytosine, and therefore a guanine got incorporated. Other spots will remain unaffected, right? So the flow cell is flushed with single species of nucleotide at a time. The semiconductor chip records changes in pH upon incorporation of complementary nucleotides with polymerase. And then the current read length that you get for this technology is 100 base pairs, and there is no imaging involved here. This is based purely on a chemical reaction and the change in pH. Now we come to long-range sequencers. Now long-range sequencers, there is one technique that is very common, and this is polymerase based again. And the challenges, and this is basically, until now, the techniques that we've studied, you need a PCR step before you can go for sequencing. This, in contrast, is a single molecule real-time sequencing. It is called SMRT. It is being marketed by a company called PacBio. Now, there are challenges to single molecule real-time sequencing. One is the signal intensity. When you do a PCR, you have a clonal amplification of your original template. So when you give your DNTPs, let's say 100 DNTPs get extended. So which means that your signal is 100 times what it would have been if you have a single molecule there, right? And therefore now you can record your signal very clearly and you can distinguish from the background. But in this case, you have only one template and therefore the signal intensity is very low. To record that signal intensity is challenging. So for example, here, how do you meet that challenge? For example, here you have a 40-watt bulb. You put it in your drawing room, you put it in your kitchen, you put it in your bathroom. The bathroom ideally would be the smallest space and therefore you'll have the brightest signal here. You get it? So what we do is we reduce the reaction volume. The space where the reaction is happening is brought into a very small volume so that while it is still a single DNA and a single nucleotide that will get incorporated, you can still record the signal. And then of course, the other important problem is the polymerase fidelity. So the polymerase is not perfect. And in fact, courtesy that the polymerase is not perfect that we are here today. Otherwise, there would still have been some small piece of DNA somewhere in the oceanic pre-ocean, right? So mistakes are important. Polymerase creates mistakes here. If you're sequencing a single molecule, you will not be able to identify whether there has been a mistake or not. So how do we, while we're still at a single molecule sequencing, how can we increase the number of times we sequence it? So for that, what we do is we circularize our template. So you add adapters at both ends. Now it becomes a circle and now you can start sequencing and it keeps adding nucleotides multiple times. So this is the same sequence coming up multiple times now. And now you can match the sequence here versus here and see what is the nucleotide that has come the maximum number of times for a given position. That would be your nucleotide of choice as compared to one odd errors that may happen. So this gives you now around 99% accuracy in the reads, right? So the solutions to single molecule real-time sequencing, the challenges were signal intensity and polymerase fidelity. You do this, you reduce the challenges by reducing the reaction. You use something known as zero mode waveguides, and I'll come to that in a moment in the next slide. And then you circularize your DNA and sequences repeatedly in the circle. So here is your SMRT, single molecule real-time sequencing. It is given by PacBio. Just a minute, yeah. It is given by PacBio. This is what is known as zero mode waveguides. This is the smallest space where DNA polymerase is immobilized. You have your template and you have your DNTPs. And as the polymerase keeps adding DNTPs, because the DNTPs are differentially labeled, you get to know which nucleotide got incorporated. This DNA is circularized so that it can be sequenced multiple times within this closed space. And therefore, what you have now is a higher read length. You can typically have around 30,000 base pairs very easily. Some of these outputs will be, output per smart cell can be up to about 10 GB and reads per smart cell can be around 400,000. And you have around half of your reads can be greater than 30 KB. Some would be even greater, around 50 KB. And then a few watt can be as high as 90 KB. Now, if you remember, I told you read length is important in assembly of the genome. The longer the read length, the lesser the chances of error. And therefore, now we are moving towards long read sequencers. And over time, Illumina and other sequencing technologies are losing out to Nanopore and PacBio. And remember, these are technologies under development. So these are still, it is not, everything is not hunky-dory and perfect. Things are still under development. But then, of course, we are slowly moving from small read sequencers to long read sequencers. All right. So the next in long read sequencers, I got stuck somewhere. Yeah. So the next in long read sequencers is Nanopore. If you see this sequencer is the size of a pen drive, you can hold it in your hand, put it on your computer and directly sequence the DNA. So this is a wonder in miniaturization. Also in terms of read length, it gives you much longer read lengths. More importantly, this in contrast to all other technologies that I've discussed till now, this is Helicase based sequencing. Okay? Now nanopores, as you know, it is a pore of nano size. This pore can be biological. I'll give you an example of that. It could be chemical, it could be a solid state or naturally found or whatever, or synthetic, or it could be a hybrid where a protein is set into a synthetic material. Right? So here is a biological nanopore. This is a protein called alpha hemolysin. It is a haptomeric protein. And if you see, this is the tunnel here. So this is your nanopore. This is the other section, transverse view of the same. So what is the idea behind this? The idea behind this is that if you set a nanopore in an electric field, there will be a certain threshold current that will flow across this continuously. If you now introduce molecules into this electric field, depending on what molecule you have introduced, there will be a characteristic disruption in the current that flows through this nanopore. For example, a smaller molecule may produce this much of effect, a larger molecule can produce even more, or depending on the stereochemistry, you could have a larger molecule which produces a slightly lesser fluctuation in the current. Fortunately for us, adenine, thymine, cytosine, and guanine produce different fluctuation intensities in nanopore. So therefore, what you do is you take your double DNA molecule, double helix, unwind it using a helicase, force it through a nanopore. You use a ratchet-like protein to force this DNA through the nanopore. And this is the position where we're recording the fluctuations in current. And the fluctuations in current are characteristic for A, C, T, and G. And that can then be digitally converted into the sequence of nucleotides. Right? This is marketed by Oxford Nanopore. Time to sequence human genome can be around 15 to 20 minutes. Size of sequencer is pen drive, but it is also scalable. You have something called as PromethION, which is an assembly of multiple nanopore sequencers put together just to increase your high throughputness. And then the cost of sequencing comes to around $1,000 here, just about $1,000. And the advantage is this is a long read sequencer. So advantages with nanopore and other long read sequencers: direct RNA DNA sequencing. It is real-time sequencing. You get ultra long reads as much as 30,000 base pairs, 50,000 base pairs, and so on and so forth. It is scalable to portable or to desktop. You can have the library preparation is very small. It takes around 10 minutes and not so complex. And then you have high fidelity or high reproducibility of the data. So if you go to this review here, this is 2018. It basically says that the ultra long reads produced by the long read sequencers, particularly the nanopore, can be the game changer in the sequencing technologies. So we'll take a small recap here. So I have told you about the sequencing technologies. Starting from Sanger, we moved to virtual terminated sequencing, which is very similar to Sanger, except that you use proprietary nucleotides where the termination can be removed after you've recorded which nucleotide got added. Then we went to Ion Torrent. Ion Torrent again is polymerase based. And here, instead of the DNTP that you add, the reporter molecule is the hydrogen ion. And hydrogen ion, basically, if a DNTP gets attached, you'll have a hydrogen ion release, resulting in loss of pH, and that can then be converted into the sequence. And you're giving only one nucleotide here at a time so that you know which nucleotide you have given. So you know at which positions you have got your nucleotide incorporated. Then we talked of single molecule real-time sequencing, where you sequence a single molecule, DNA polymerase is immobilized in a very small space, and then you sequence it the way you normally do with other sequencing techniques. And then we have talked of nanopore. Nanopore is a helicase-based sequencer. So in contrast to preparing a complementary strand, here you directly read from a single strand what is the nucleotide that is passing through a nanopore, right? And SMART and nanopore are long-range sequences. Illumina and your Ion Torrent are short-range sequences. Now we come to NGS applications. Now, why NGS has become so important in biotechnology is because there is nothing that is untouched by next-generation sequencing technologies. For example, you can have a DNA sequence, so you can use it for variant calling, SNP calling, or structural variations. So this is called DNASeq. Then you can use it for studying the epigenome or the methylation status of the genome. This is called BSeq or bisulfite seq. Then, of course, you can do transcriptome analysis, where you are typically studying the entire mRNA of a That's called RNASeq. And then you could study DNA protein interactions, which is called ChIPSeq. And again, this is where in case you have proteins that bind to in a sequence specific pattern manner, then you can use this technology very clearly to understand what is at what time and what is the impact of the DNA protein binding on the expression levels of the genes and so on and so forth. So your analysis can be depending on what is your experiment. So the key features of NGS, scalability, as I told you, it is massively parallel sequencing, high throughput, parallel processing of thousands of samples. Speed, it is much quicker than Sanger sequencing. You can do a whole genome in 2 to 3 days or maybe 20 minutes to 30 minutes. Then you can do an entire exome in a day. I'll come to what is an exome. It is high resolution. It is deep. You have a lot of depth in sequencing. So you're very sure when you say that there is a polymorphism here, you're sure of what is the extent of polymorphism and you're sure that you're not going wrong here. I told you it is high coverage, single base pair resolution. And then the assembly that you do most commonly here is reference-based assembly. You already have a sequence genome. You just line your reads up there and get your sequence. As of now, with long read sequences, you can also do a de novo assembly. People have attempted de novo assembly with small read sequencers as well, but then that is not as successful. So exome sequencing. So as I said, while the cost of genome sequencing has come to around $1,000, somewhere around $1,000, not exactly $1,000, it is still prohibitive. $1,000 today would mean around how much? 70,000 rupees or something. And we know that most of the disease-causing mutations are within the protein coding genes, which are only 1.5% of the entire genome. So if 85% of the mutations that cause disease are within the protein coding genes, I can do only the sequencing of the protein coding genes and leave aside the rest of the genomic material. And that would bring down the cost of sequencing very heavily and still not compromise too much of the information because you still get 85% information of your disease susceptibility. So that is what is exome sequencing. You can basically fish out the regions that code for genes using probes and then directly do the sequencing of exactly only those regions instead of sequencing the entire genome. This brings down the cost and it is extremely useful in whole genome analysis of biomedical conditions. Some of the Mendelian diseases that need via next generation sequencing, you have a host of them. I'll not go into the details. So this is providing an improved clinical diagnosis, more accurate genotype-phenotype correlations, and new insights into the role of rare genomic variations in disease. You can do targeted panels. Suppose you know that these mutations at these positions in the human genome are important for a certain type of disease, let's say cancer. So you can only sequence these regions instead of sequencing the entire genome. So you can select a set of genes or a set of positions that you want to sequence. Multiple genes can be assessed across many samples in parallel, saving time and reducing costs associated with running multiple separate assays. And this is basically also important because it produces a smaller, more manageable, and more significant data as compared to a whole lot of background that you may get when you do a whole genome sequencing. Now, one of the important things that has been facilitated by next generation sequencing is metagenomics. Metagenomics is also known as community genomics, where you can study the entire, or environmental genomics, where you can study the entire content of microbes in a given sample without any loss of biodiversity. Normally, what we did before the NGS came into the picture was first let's say you have to identify the biodiversity in the soil around Hindu College. So you would get the soil sample, you would then do a culture, grow the bacteria, and then whatever bacteria you recover, you'll isolate the DNA and then sequence. Now the problem with that is that when you grow the culture in lab conditions, a lot of biodiversity is lost and it is never recovered in your final set of population that you retrieve after culturing. So with NGS, what you can do is directly get the environmental sample, extract genomic DNA directly, do a PCR of the hypervariable region, sequence, and then do a bioinformatic analysis. Or you can do a whole genome metagenome sequencing. You can directly sequence the entire genome and then look for what it matches to in the databases, right? So it is basically genomics on a huge scale. It enables to survey different microorganisms present in a specific environment, such as water, soil, etc., to be carried out. Also, you can identify community relationships. Suppose A type of bacteria is present in a very high proportion, let's say 90% as compared to a B type of bacteria. So you know what is the dominant population in your community versus the other population in the community. And that can also allow you to identify the functional relationship between communities in a given ecosystem, right? And why do we want to identify new microorganisms or characterize new microorganisms than what we already know? So as you know, we want new molecules for therapeutic intervention, because for most of the existing drugs, the pathogens have now developed antimicrobial resistance, so AMR. So basically, for any drug that you give now, the pathogen is also already resistant to the drug, and it is now a big problem. So we now need new set of molecules that can provide us new interventions. And for that, you first need to identify the uncultured microbial diversity that exists today. So metagenomics, 16S RNA typing. So 16S RNA region is characteristic for each species. You can do your 16S RNA targeted sequencing of 16S RNA genes in the population that you have, and then identify the species, the number of species that are present in your sample, and also their community relations based on the abundance of a particular type of 16S RNA signature. Right? So there are some microbiome projects that are very much running as of now. So you have the human microbiome project. You would have heard the gut microbiome project. There we are trying to now identify the total diversity that exists in the gut of human beings. And the idea is that it has been postulated that healthy human beings will have a different set of organisms in their gut microflora as compared to unhealthy human beings. So a lot of disease that you now see are being associated with the gut, the composition of your gut flora and fauna. And therefore, now we are trying to understand the gut microbiome in a lot more details. You have Earth microbiome project. We are trying to systematically and catalog the entire micro population that exists on Earth. And then also in the environmental para space, so as to define the protein universe and the community structure that exists. We are also into pharmacogenomics. So pharmacogenomics is the study of the genomic influence on drug response, often using high throughput data. So high throughput data means NGS or next generation sequencing. I'll give you an example of how this is done. For example, there is a purine analog that is used in cancer studies. So this is called 6-mercaptopurine. It is cytotoxic and immunosuppressive drug. 6-mercaptopurine is given as azathioprine. It is metabolized to 6-mercaptopurine in the body. 6-mercaptopurine is further metabolized in the body to finally give the functional product, 6-thioguanine nucleotides. 6-thioguanine nucleotides, when incorporated during DNA replication, actually kill the cells. However, the 6-mercaptopurine that you're administering can also be metabolized by another enzyme in the body that is known as TPMT, or thiopurine methyltransferase, which adds a methyl group to mercaptopurine. And now this molecule, 6-methylmercaptopurine, is dysfunctional, which means it will not have the lethality that we want here. So this is our functional molecule. This is our side molecule, which gets converted via TPMT, right? So 6-mercaptopurine is a cytotoxic immunosuppressive drug. This is the structural formula for that. It is used to treat lymphoblastic leukemia, autoimmune disease, inflammatory bowel disease, post-solid transplant. The cytotoxic end product of 6-mercaptopurine is 6-thioguanine nucleotide, which interferes with DNA replication, leading to cell death. However, as I told you, 6-mercaptopurine can be converted to 6-methylmercaptopurine. Now, this molecule is useless for us. So what we want is that most of our medicine should go this way and not this way, right? Now, if you see the TPMT is polymorphic, and there are each individual can have variations in the DNA that allow for a slightly lower efficient TPMT molecule to be present. For example, here you have this variant here where you have guanine replaced by cytosine at 238 position, resulting in the replacement of alanine by a proline. And this molecule is less effective as compared to the wild type molecule. Likewise, you have other variations. So basically, TPMT is polymorphic in the human population. And therefore, depending on whether the individual is wild type or variant for TPMT, the dose may be toxic or less toxic. So the TPMT activity for variants, which is homozygous variant, is very low. It is slightly higher in heterozygous individuals, and it is very high in wild type. Right? Now, why this is important is because when you give the dosage, normally what happens is when you go to a doctor, the doctor says, okay, sabka wahi roj ka hai, take these medicines, one in the morning, one in the afternoon, one in the evening. But then this is not customized medicine. This is basically a blind thing that you are giving the same dosage to every individual, irrespective of what variations he is carrying. Now, in case of TPMT-C, if what happens is, if this is your regular dosage for both variant, heterozygous and wild type, homozygous variant, heterozygous and wild type, you will have a lot of toxicity in the variant because all of it is available for cytotoxicity. It will be slightly less in case of heterozygous variants, where while it will still be toxic, but it will be least toxic in a homozygous wild type here, right? So what basically this indicates is when you go to a doctor for a medicine, you have to know what polymorphisms you carry. Based on that, the doctor can suggest you the dosage that will be optimum for you without producing excessive toxicity. For example, here for an individual who is variant homozygous, where there is no TPMT activity, which means all the other thiopurine that is given directly goes into 6-thioguanine. These can be administered a very low dose to be as effective as somebody who is wild type, where most of it is going here, and therefore a higher dosage has to be given for the same effectivity, right? So basically, so this example very clearly indicates that based on individual variations, the dosage variation of the medicine is important, which we have not been doing until now. And therefore you hear people, some say that the medicine was very effective for me. Others say it was absolutely, it actually caused a reaction to me. So that is basically because we are all different. And because we are all different, the medicine interacts with the genome differently depending on what variations you're carrying. So this is one very good example of that. Now coming to genome sequencing milestones. The first sequence was sequenced way back in 1977, bacteriophage 5X74. Then, of course, Haemophilus influenzae, and since then there have been continuous achievements. And of course, the crowning glory was human genome sequence in 2001. And now we can also sequence DNA from pre-existing animals or mammals. For example, Neanderthal, which is our closest human relative. A part of this, its DNA was resynthesized from the fossils that were isolated from Heidelberg, I think. Then this is a good review that you can see for applications of DNA sequencing at 40, past, present, and future. I'll also give you a DOI in some of the slides that is there. Then recently, we have also sequenced the axolotl genome. This is the largest genome to be sequenced till date. It is 10 times as large as the human genome. So this is the largest genome ever sequenced. Scientists have decoded the genome sequence of axolotl, the Mexican amphibian. The human genome size is 3.2 billion base pairs. It is approximately 32 billion base pairs, which makes it 10 times the size of the human genome. And we are interested in seeing why there is so much of excessive genome size in these individuals. And if you know, there is something called C-value paradox, which states that the genome size has no direct correlation with the complexity of the organism. There is one such example of that. Then all next generation sequencing file formats are standard. So you'll have 4 lines in the sequence. The first line would be your basically read ID. Then you have the sequence. You have a repeat of read ID. This is optional. And then you have a quality score or the quality call for each of the nucleotide here in the sequence. So this four-line format is standard for all NGS data. Then, of course, because you get reads that have to be aligned for to a different genome for assembly, depending on which platform you're using, you have platform-specific aligners, and then you have, for example, Solid, you have very specific platforms, 454 platforms. Then you have some platforms that can take reads from any of the sequencing platforms. For example, you can have Solid, you can have Burrow-Wheeler's algorithm. You can both Solid and 454 and Illumina data can be used here. So some algorithms are specific to a sequencing platform. Some algorithms are generic and can take reads from any platform and align it to your reference genome. I've already given you this reference, DNA sequencing at 40, past, present, future. It is a very nice read, and the first few lines are very interesting, where they say that to sequence initially, it took 3 months for one very small stretch of DNA. And now we're sequencing the entire human genome in a matter of less than half a day. Now, from your point of view, sequencing mostly is done by companies. So labs may also do it, but mostly now it is outsourced because it is a very standard practice. So it is mostly outsourced to companies or CSOs. Now, the part where the student role comes in is in the NGS data analysis. Now, NGS data is typically big data characterized by velocity, veracity, and variety. And you need to have certain data on data analytics and computational skills to be able to analyze this data. Very importantly, you must know one programming language. Python is the most preferred as of today because most of the algorithms that are being used now are written in Python. Then R is equally important. R is open source, therefore you don't have to pay anything for this. So this is very important. It is a statistical language, and it comes in very handy when you're analyzing your data. You have to be good at biostatistics because we are dealing with high throughput data, which means there is a lot of data, and from this lot of data, you have to pull out significant data. So at every step, there is a statistical test that has to be put in. And therefore, you must know a bit of bioinformatics so as to be able to analyze the data. Awk is a very handy language that can be used for analyzing data, provided it is in columns and rows. And all these are not very difficult. Actually, it basically is on interest. Then there are some very fixed tools that are used for data analysis, NGS data analysis, for example, Picard, SAMtools, BedTools, Bismarck. Bismarck is specifically for bisulfite-seq data, which I've used during my postdoc. And then, of course, you need familiarity with the Linux environment so as to ensure that you can work comfortably and quickly and manage this size of data in the stipulated time that is given to you. Then, if you want to learn R interactively, you can go to my site here. This is called Vipin C Classroom. If you Google Vipin's Classroom, it will take you directly to my page. And you can go to Vipin's Crackets. And here I have given some instructions how to learn R interactively and how to download and install R. This will give you the basic introduction to R, and then you can move on and do as per your interest. Programming is basically intoxicating if one gets into it. Some people may like wine, some people may not like wine, so it is up to your taste. But if you get into it, this is the best area to be in if you want to be recruited with good companies. It gives you a lot of IT skills and everything. So you can go into IT as well as into bioinformatics. And I would suggest that every student should know some basic programming now, because now the data is not one gene or two gene or three gene or one protein or two protein. It is the entire proteome, the entire genome, the entire transcriptome. And unless you know a bit of programming, it will be difficult to analyze the data. So you can have a good start here. You can start with R, and programming is easy. If I can learn it, then you can definitely learn it, right? All right, so we started with Sanger sequencer here. This is the ABI present sequencer. Then we went to Illumina, we went to Ion Torrent. Then we went to a SMART or single molecule real-time sequencing. then we went to Nanopore, and then we went to Promethan, which is Nanopore multiplexed, right? So I'll stop here, and I hope I've been able to do justice to the topic that was given to me, and I am open to questions if anybody is interested in asking. These were some of the early birds who came very early in the talk, right? So I took some screen grabs. Thank you for your attention.
Thank you, Dr. Vipin, for providing such exhaustive and